Okay, welcome to this talk for DGPPN Kongress 2022 in Berlin. I want to thank the organizers for inviting me to take part in this symposium. And today I want to talk about structural and functional MRI and how they are providing complementary information when we are investigating neurocognitive aging. So I have no conflicts of interest, financial or otherwise, to declare. And I want to start with a brief introduction into neurocognitive aging, in addition with a description of how we are investigating this using neuroimaging. Then I want to go through all these modalities of magnetic resonance imaging step by step and discuss what information they provide us with about the neural correlates of cognitive abilities in old age. So although cognitive performance usually declines with increasing age, as is here shown for episodic memory performance, some older adults are known as successful agers, which means that they have memory performance comparable to that of young subjects or even better than that of young subjects. It is still unknown why successful agers are so successful in terms of their cognitive performance. And we want to especially investigate the neural underpinnings underlying cognitive performance, especially memory encoding and retrieval in old age. So we measured brain activity while subjects were looking at photographs, and these photographs were either new to them at the time of presentation, so novel images, or they had been pre-familiarized. And the subjects had seen them a couple of times before the experiment, so-called master images. After the experiment, in a subsequent memory test, subjects are then being presented with all the old images from the encoding session, as well as a couple of new images. And they are supposed to rate them on a 5-point Likert scale, ranging from 1 for this item is definitely new to me, up to 5 for I've definitely seen this item in the encoding session. So this experiment is called the FATE paradigm, and it is used as the prime functional MRI experiment in the DELCODE study run by the German Centers for Neurodegenerative Diseases across Germany. So that's a multi-center study focusing on AD risk states such as mild cognitive impairment and subjective cognitive decline. We are in the lucky situation of having also measured healthy young and older adults using this fMRI paradigm. This is the Autonomy in Old Age project at the Leibniz Institute Magdeburg. And here we have at the moment 106 young subjects aged 18 to 35. And we're using these young subjects as a reference group when looking at the older adults. And we have 111 older subjects aged 60 plus, and they are, in terms of the age range, exactly equivalent to the healthy controls from the DELCODE study. There are a few other subjects which I won't discuss in this talk, and all of these subjects were scanned using the exact same protocol as in the DELCODE study, which is very advantageous for our analysis. So results from this dataset were first reported in this 2021 neuroimage paper, in which we used Bayesian model selection to identify the optimal modeling of first-level fMRI data. And we came up with a model which includes a novelty effect and a non-linear transformation of the subsequent memory response. Using this model, we are then inferring on effects of interest in the functional MRI data. So using this model, we are looking for effects of novelty processing by comparing novel images with master images. This is here shown on the left side. And we infer on subsequent memory effects by contrasting remembered items with forgotten items, which is here shown on the right side. So if we look at the age-related differences between young adults and older adults with respect to these contrasts, we see that both contrasts actually have very similar age group differences in the sense that we have reduced activations in the older adults in regions such as hippocampus, parahippocampal areas, and other temporal areas, whereas we have reduced deactivations in regions of the default mode network, such as the precuneus or anterior cingulate cortex. So this motivated us to compute two what could be called fMRI biomarkers. We usually call them single value scores, which are quantifying the deviation of a given older adult from the reference group of young subjects. So both of these measures start with a given contrast in the young subjects, can be novelty or memory. And first, we're identifying those boxes where this contrast is significantly positive or significantly negative, here in red and blue, within the sample of young subjects. Then we're taking this voxel and calculate the so-called FATE score for functional activity deviation during encoding. This was introduced already 10 years ago. And this score basically subtracts the average effect within the voxels with positive effect from the average effect in all other voxels. Here, we developed an improved version of the FATE score called the same score for similarities of activations during memory encoding. And the same score goes beyond the FATE score by A, operating directly on the fMRI activity levels, B, accounting for reduced activations as well as reduced deactivations in older adults, and C, by normalizing with the variability of the young subjects, which are used as a reference sample when computing those scores. So typically we are computing both scores, FATE and same, for both contrasts, novelty and memory, which then gives us 4 indices for each subject. And when we compare these scores between young and older adults, we find that both scores significantly differ between young and older adults when computed from the memory contrast. And the FATE and the same score also does so when computed from the novelty contrast. When we are controlling for age group, that is when we are correlating those scores with other indices of neurocognitive aging within age groups, we find that both scores are significantly correlated to memory performance. This is here shown in the second row, but they are not correlated with hippocampal volume in neither hemisphere on neither contrast using neither of the scores, which was quite surprising for us. So these results were published in a human brain mapping paper from last year, where we presented this method of reductionist scores computed from whole brain fMRI contrasts, which are somehow indicative of, let's say, disturbances in memory encoding fMRI activity. So with that, I want to come to the role of structural MRI. And I said in the beginning that there is this phenomenon called successful aging. And it is yet an open question whether successful aging is due to a brain structural integrity, that is the fact that these subjects simply have more brain tissue available, which would express itself in structure MRI and could be termed the brain maintenance view, or B, whether successful aging is due to more efficient information processing in those successful ages, which would then express itself in functional MRI patterns and could be termed the cognitive reserve hypothesis. So it is clear that we will not be able to decide between those two theories today, but we can note that different hypotheses about successful aging make different predictions about the correlates of memory performance, because according to the brain maintenance view, differences in memory performance should co-occur with differences in structure MRI patterns, whereas according to the cognitive reserve hypothesis, differences in memory performance should correlate with differences in functional MRI responses. So we actually have the data available to test these hypotheses, But before we do so, let's pause for a moment and summarize what we have. We have 2 fMRI contrasts indicative of novelty processing and subsequent memory. We have 4 fMRI scores computed from these 2 contrasts, paid in the same score. We also have the behavioral data available, that is, how often each subject responded 1, 2, 3, 4, 5 to old versus new items. Additionally, from each subject, we measured a structural MR image from which we calculated voxel-wise gray matter volume. And finally, each subject performed a resting state fMRI session from which we calculated voxel-wise mean percent of amplitude fluctuation, or M/F in short. So the idea here is to use all of these feature sets separately to predict some target variables of interest, namely age group, that is whether the subject is young or old, chronological age in years, and memory performance measured as area under the curve in the subsequent memory test belonging to the fMRI paradigm. Now, first, we observe that age group can be classified based on all these variables. And here we already observe a clear hierarchy from behavior over functional up to structural information, meaning that nothing outperforms gray matter volume when classifying age group. If we are looking at the continuous variables, and here only focus on the older subjects, then we see that chronological age is again best predicted from structural MRI, which is here shown in green on the right side. However, gray matter volume has absolutely no predictive value for memory performance in the older subjects, which instead is better predicted from functional MRI contrasts, here shown in blue on the bottom right. So these results were summarized in a recent eNeuro paper in which we found these that structural and functional MRI data, no matter how they are represented generally, have these complementary roles in terms of what kind of indices of cognitive aging they are correlated with. Structural being more correlated to the biological measures such as age, and functional MRI being more correlated to the psychological measures such as memory performance. So one can look at these data from a different perspective. And here I want to bring the fMRI scores back into the game because if we are looking again at the plot which I just showed you, you probably also noticed that the single value fMRI scores outperform the whole brain fMRI contrast maps when predicting memory performance in the older subjects. So this means that just using 4 features, we are able to better predict memory performance than using the whole brain fMRI contrast maps with information from all individual voxels. This is also true for when predicting independent measures of memory performance. So when we're predicting memory performance in completely different neuropsychological experiments, we here use the verbal learning and memory task, which is shown on the top panels, and the Wechsler memory scale shown on the bottom panels. And we can see that the feature set comprising all the 4 fMRI scores is the only one which achieves above chance prediction accuracy for all of these measures of memory performance. One can look at this data set from yet another perspective when focusing more on the resting state fMRI. If we look back to this accuracy plot for age group classification, we see that the performance of resting state fMRI is kind of between the one for structural and the one for task-based functional MRI. This is also the case when predicting chronological age as a continuous variable, and when predicting memory performance as a continuous variable, resting state fMRI is very similar to gray matter volume. So we wanted to follow up this distinction between age and memory, as well as structure and function MRI. And in order to do so, we did a subgroup analysis within the older subject and partitioned those older subjects either based on chronological age, resulting in young versus older subjects, or based on memory performance, resulting in subjects with lower versus higher memory performance. What we then found is that there is a double dissociation between memory versus age on the one hand and functional versus structure MRI on the other hand, in the sense that when we're partitioning the older subjects by age, there are no significant effects with respect to functional MRI, but strong differences in gray matter volume. Whereas if we are partitioning subjects by memory performance, there are no significant effects in structure MRI, but robust memory-related brain activity differences. Now the question, which camp does resting state belong to? Well, the MPRF maps behave just like the GMV maps, meaning that the informational content of resting state fMRI is closer to that of structure MRI than to that of task-based function MRI. Okay, finally, I want to give a brief outlook to the clinic, because we have already applied this methodology of the reductionist fMRI scores to the first Delcode data that we have received. And I want to just share two very interesting first preliminary observations that we did with you today. So the first thing that we noticed is that when we calculate the FATE and SAME scores for the different diagnosis groups in the Delcode study, we see that the scores from the memory contrast, which are at the bottom, distinguish MCI and AD in orange and red from all the other groups, whereas the scores computed from the novelty contrast distinguish MCI and AD from each other. So this is a very promising result in the sense that those scores might at some point allow us to derive a proxy for Alzheimer's disease risk state. Finally, there are no effects of ApoE genotype on the scores in any of the group, except for the relatives of the Alzheimer patients. So in the AD relatives, we in fact see significantly different scores for E3 homozygotes when compared to carriers of the E4 allele, which is usually considered as a risk allele for Alzheimer's disease. So to sum up, we have introduced two fMRI biomarker scores, or single value scores, which hold rich information about memory performance and other cognitive abilities in old age. Structural MRI is best at predicting chronological age, but memory performance can only be predicted from functional MRI, which more strongly supports the cognitive reserve hypothesis of successful aging. And finally, single value scores significantly differ as a function of Alzheimer's disease risk state, which is a promising result for further clinical investigations. So with that, I want to thank my collaborators, Björn and Anni at the Leibniz Institute in Magdeburg, and additionally, Jasmin at the DZNI Göttingen. And I want to thank you. Are there questions?